part of Chapter 13 will be covered on your own by reading the first few sections of Chapter 13 and completing an online worksheet. This worksheet is set up to essentially be a quiz, but it is not timed, so you'll have unlimited amount of time. You do have one submission though, so take your time in completing it. Make sure that you've got those answers correct before you submit it. I would encourage you to read through your book and those assigned sections thoroughly. Make sure that you get all the way to the end before you complete those questions so that you get the points, the full point values for each of those questions. This section covers chemical and physical agents that we can use to control microbial growth. It goes over the true definitions of some common words that we've probably heard before, things like sterilization, disinfection. So that stuff is pretty straightforward. I would like to have you complete that digital worksheet, which can be found under the quizzes tab or within the weekly module. Make sure that you do that before you proceed with this particular lecture video. Lecture video A is going to cover slides 1 through 9, and then lecture video B, slides 10 through 23. So if you've got that worksheet completed, let's begin. We are going to be looking at ways in which we can eliminate pathogens from the body as we move forward. So the term antibiotic is a term that refers to a compound, a substance that is made by one microbe and is kind of put out in the environment with the idea that that compound is going to selectively kill competitive microbes within the area. If, as a species of bacteria, you can make an antibiotic to kill off your competitors, that's a good thing for you to have. So many of our first antibiotics were isolated from microbes that made them naturally. We talked about one example of this when we dealt with the penicillium mold. This was our first source of a natural antibiotic. In that particular instance, our chemotherapeutic agent that we now call penicillin was made and inhibited the growth of bacterial species. So we can have chemotherapeutic agents, and that term chemotherapeutic, oftentimes we associate that with like cancer treatments. The term chemotherapeutic means a chemical therapy, okay? So it's much broader than what we use it in normal day speak. So be aware of that. But we can take these antibiotic compounds once we've identified what they are, and sometimes we can synthesize them within a lab. So when we start looking at these compounds that are going to be used to target pathogens within the body, one of the most critical ideas that we have to develop and consider is the idea of selective toxicity. Selective toxicity refers to inhibiting pathogen growth. Most commonly, it's going to be a bacterial species, but we do not want to harm the host cells. So when we talk about selective toxicity, this is a critical feature that all of our chemotherapeutic agents need to have. We don't want to give a patient a drug, even if it's great at killing bacteria. Well, if it hurts that patient's cells, that's simply not going to work for us. So the idea of selective toxicity, inhibiting a pathogen without harming a host cells, is very, very important when we're developing drugs. It's important to note that there are multiple types of drugs that are available, and there is no one drug that will affect all microbes the same. So when we start looking at different categories of pathogens that we might be inhibiting, we use different names to reflect those different types of drugs. So when we start talking about antifungal drugs, those are drugs that are going to inhibit fungal growth. Antiviral drugs are drugs that inhibit viral replication within host cells. Antibacterial drugs are now synonymously used with the term antibiotics. So as we proceed from here on out, antibiotics are going to be the term that we use when we're talking about drugs that specifically inhibit prokaryotic bacterial cells. So when we consider these three broad categories of drugs and the organisms that they inhibit, we need to think about the idea of selective toxicity. So the activity that we have there, first do no harm, can be completed on a scratch paper if that's all that you have in front of you. But I would like you to take a second, think about selective toxicity, think about what you know about fungal species, viruses, and bacterial species. Which of these three groups of chemotherapeutic agents is going to be most difficult to develop drugs for because selective toxicity is going to be difficult to achieve? On the other end of that spectrum, which of these three is going to be most easy or easiest to develop while still maintaining selective toxicity? Pause the video if you need a second. Selective toxicity tells us that we have to target the pathogen without affecting the person who is taking the drug. Well, start thinking about the different components of a cell that we would be targeting. The most difficult types of pathogens to develop drugs for are viruses. We don't have a lot of antiviral medication. Why is that? Because viruses rely on our cell machinery to replicate. And so when we look for a target, something that is unique to the virus that we can disrupt, but not hurt our own cells, there are a limited number of things that we can target in viruses simply because they use so much of our own machinery. So which of these guys then would be the easiest to develop selectively toxic drugs for? Those would be our antibiotics. So our antibiotics or our antibacterial drugs are going to be targeting prokaryotic cells. Our cells have a lot of differences between them in terms of structure, in terms of various processes, and so we can target those things that are unique to our prokaryotic bacterial cells without harming our own cells. Antifungal drugs fall in between, and that's because fungi are eukaryotic. They're going to have a lot of the same structures and organelles that they're going to be using. So when we start talking about these different types of drugs, and we're going to focus primarily on antibiotics, selective toxicity needs to be playing in the back of your mind. All right, so when we look at antibiotics, we can classify those antibiotics as either broad spectrum or narrow spectrum because their spectrum of activity, or the number of microbes that they kill, is different. So the spectrum of activity of a narrow spectrum antibiotic is small. There are a limited number of microbes that a narrow spectrum antibiotic can affect and harm or inhibit in some way. So an example is penicillin. Penicillin is a compound that targets the cell wall structure. We have talked about how penicillin comes in and binds to the active site of the enzymes that are assembling that cell wall. Well, the cell wall is exposed in gram-positive cells, and so penicillin can effectively inhibit gram-positive cells much, much better than gram-negative cells. Gram-negative cells, you'll recall, have an outer membrane that protects their peptidoglycan from exposure to that penicillin compound. Ampicillin is a modified version of penicillin that has a broader spectrum of microbes that it can kill. Ampicillin has the ability to pass through that outer membrane and get down to that thin layer of peptidoglycan where those enzymes are busy assembling that cell wall. So ampicillin is a broader spectrum drug because it will kill not only gram-positive bacteria, but it can kill gram-negative bacteria as well. So when we talk about antibiotics moving forward, this is something to kind of keep in mind, whether we're dealing with a broad spectrum versus a narrow spectrum antibiotic. So when we have a patient who comes in as a caregiver, they're kind of having a balancing act between wanting to treat that patient quickly, but wanting to treat that patient who has a bacterial infection with an antibiotic that is effective at killing off the pathogens. So the trade-off here is really time. If a caregiver decides to go with a more narrow spectrum antibiotic, there's a limited number of cells of bacterial types that drug is going to target. If the caregiver chooses to go with a broader spectrum, well, that may be a better choice seemingly in the short run because you're more likely to find a drug that has an effect on that pathogen. But broad spectrum antibiotics can sometimes have collateral damage, if you will. Oftentimes these antibiotics are more damaging to our normal microbiota, and so sometimes they can have a greater risk of diarrhea or some of those other opportunistic infections that we talked about. But the trade-off is that sometimes identifying which narrow spectrum antibiotic is best takes a little bit of time and a little bit of testing. So there's a balancing act here. We're going to begin by looking at how we can learn about antibiotics in a lab setting. So one of the most important features of any antibiotic that we'll test is what is called the minimum inhibitory concentration, or the MIC. So in this particular instance, we are looking for the lowest concentration to prevent growth. Now, we may have a drug that has a minimum inhibitory concentration. We won't know whether or not it's bacteriostatic or bactericidal, but we just know that it inhibits, that concentration of antibiotic inhibits the growth of that bacteria. So when this is done in a lab, it is limited to one antibiotic and its effects on one species of bacteria. So this MIC is going to be different among different microbial species if we're looking at a single drug. If we are looking at multiple drugs and how they affect a single species, each of those drugs is going to have its own MIC as well. So let's look at one example and see how this is set up. So in this particular instance, we have tetracycline, and our unit of measurement in our media is micrograms per milliliter. And so what we have done is we have inoculated this particular media with our antibiotic, 8 micrograms per milliliter here. And then we have done what is called serial dilution. That means that we take and we cut the concentration of that drug in half, and in half, and in half. And we end up with very small amounts as we get towards those smaller concentrations. So then after we have our entire media set up, we take the same amount of bacteria and we inoculate all of those tubes with the same amount of cells. And then we let them grow up overnight in an incubator. And this is what we see. You'll notice at the end down here where our concentration of antibiotics are low, we have cloudy media. What does that tell us? Well, it tells us that there was bacterial growth. There was not enough antibiotic here to inhibit the bacterial growth, and so those cells underwent binary fission because they were unaffected by that concentration of the drug. We see that the tubes over here on the right, the media remains clear. Well, what does that tell us? It tells us that no bacteria grew. We didn't have binary fission taking place. It does not tell us that those cells are dead. That's an important feature. We are just identifying the inhibition point. So the point, the first concentration that we have that shows no growth, so therefore the broth is clear, that's our minimum inhibitory concentration. So for this species, for this drug, 1 microgram per milliliter of tetracycline is the minimum inhibitory concentration. But in a clinical setting, this isn't practical. You don't want to just limit yourself to testing one drug. So that's a major limitation of this type of experiment, but in lab, it's very useful. So let's look at another way in which we can determine the minimum inhibitory concentration. So what's depicted over here on the right is what is called an E-test. So this one, again, is going to help us to identify the minimum inhibitory concentration, but this one is done on an agar plate. So we take a petri dish, and we have our species that we're interested in testing. We create what is called a lawn of bacteria. So you will inoculate the plate by taking your loop with bacteria and spreading it all one direction, back and forth, coating as much of that plate as possible. And then you'll turn it 90 degrees and you're going to do that same movement. The idea here is to coat the entire surface of the plate with bacteria. And then you take one of these manufactured strips called an E-strip and you lay it on top of your inoculated plate. So it's going to lay right on top of that bacteria. And then you incubate it. During that time of incubation, 2 important things happen. One is that the bacteria that has the capacity to grow and undergo binary fission is going to do so. And we can see that growth kind of out and around the edge here. That's what that white is. It's bacterial growth. But as that plate with the E-strip is sitting in the incubator, there is an antibiotic that is infused within that strip. And so that antibiotic is going to diffuse from the strip out into the media. And there are different concentrations that you can see. We have high concentrations up here. And as we get farther down our E-strip, we're going to have our concentrations decreasing and decreasing and decreasing until there's nothing. What that means is that the concentration of antibiotic in our media is going to be high up here, and it's going to decrease and decrease and decrease and decrease until there's nothing down here. What we see around our E-strip where the antibiotic is high is that the antibiotic has inhibited the growth of the bacteria. So we call this area of clearing the zone of inhibition. So this is where the drug acts to prevent our bacteria from undergoing binary fission. Again, we cannot tell if the bacteria is dead or if they simply aren't dividing. But this again allows us to identify the minimum inhibitory concentration, where our zone of inhibition meets the growth on that E-strip is where we have our minimum inhibitory concentration. So the E-test will speed up this process, but again, it's in a clinical setting. You're still looking at one drug and one species of bacteria. So we have a method that we developed to test multiple antibiotic discs. Each disc is going to have a different type of antibiotic on it. So we're still working on this principle of having antibiotic on something we placed on our media, and it's going to diffuse out into the media. It sets up our concentration gradient where our antibiotic close to that little disc is very high, and we get less and less concentrated as we move away. So here we use little filter papers that have different antibiotics. So we set up our lawn of bacteria, again inoculating one way and then 90 degrees and inoculating again. We then place our discs in a very specific pattern using a specific piece of equipment. Here, and I'll show you that. Here, this is a little bit easier to visualize. So this is going to be our disc dispenser, and each of these tubes is going to hold a disc. The Kirby-Bauer method is done on a very specific type of auger so that this test is standardized across all labs. So after we inoculate our bacteria one way and another and we get this nice lawn, we take our disc dispenser and we set it over our plate, and then we push down that plunger, and simultaneously all discs are going to fall in a nice evenly dispersed pattern on that plate. We'll cover the plate and incubate it, and then we will look at the zone of inhibition. So this is not quite as easy as looking at the zone of inhibition and going, oh, well, this is the biggest one. That must be the best antibiotic. We measure them, we measure the zone of inhibition, and then we compare it against a standardized chart. So we do want to look at the size of our zone of inhibition, but that's not the be-all, end-all. There's some more measuring that has to go on. But what I'd like to show you here is how we can look at and compare different antibiotics and their different effectiveness against a species of bacteria. So let's look over here at first. This is a plate that has been inoculated with a lawn of Staphylococcus aureus bacteria. I don't care that you know any of the specific antibiotics that we're looking at, but what you'll notice is that this particular species of bacteria is susceptible to many of these antibiotics. We see a zone of inhibition, this clearing around many of our antibiotics tells us that those antibiotics are effective at inhibiting our bacteria. We say that the bacteria is susceptible to their effects. What I'd like you to look at, and what we're trying to demonstrate in your textbook here, is the OX antibiotic disc has a zone of clearing. This zone of inhibition is wide over here. When we look at this exact same antibiotic disc over here, notice that there is no zone of inhibition. This type of bacteria on this plate is resistant to the effect of that antibiotic. So this particular species of bacteria is called MRSA. That acronym stands for methicillin-resistant Staphylococcus aureus. So this is a special type of Staphylococcus aureus that has evolved resistance to the effects of one of our antibiotics. It is no longer inhibited by that antibiotic. We see zero zone of inhibition. So whereas it was susceptible, we have undergone mutations and now it is resistant. We will talk about how those mutations arise, but methicillin-resistant Staphylococcus aureus is a type of bacteria, a type of Staphylococcus aureus, that is no longer inhibited by many of the drugs that used to be effective against it. So that's what they're trying to show you here. All right, so here let's click back real quick. So we've said that at this point, none of these drugs are going to be able to, or none of these tests are going to tell us if our drug is going to kill our bacteria. So if we want to know the bactericidal concentration, we have to take these steps just a little bit farther. So what we can do to identify the minimum bactericidal concentration, the MBC, is that we can take any of those tubes in our dilution that had no growth in them. So remember, these were the ones that had a sufficiently high concentration of antibiotics that nothing grew, but we didn't know if the cells were dead or if they just weren't capable of dividing. So we take those tubes and we spin them down in a piece of equipment called a centrifuge. So in a centrifuge, you put tubes in at different angles, and then the machine spins them very, very fast. What that does is it forces, using centrifugal force, which is why it's called a centrifuge, it forces all of the cellular debris to the very bottom of those tubes. Now, it might be living cells, it might be just dead debris, we don't know. But after we centrifuge and spin down those tubes, they will have a pellet at the bottom. It'll be a tiny little white spot where we can have our cells. Again, they may be living, they may be dead, we don't know. So what we do is we pour off the antibiotic-laced media, and then we take just that pellet that now has no antibiotic contacting it, and we transfer it to fresh media. So now, if the cells are capable of dividing, if they were alive, then they will begin to grow, and we'll see this fresh antibiotic-free media becoming cloudy. That tells us that they were still alive if they can grow in that fresh media and make it cloudy. Now, if we take those no-growth tubes, we spin them down, we give those that pellet some new fresh media and it doesn't grow, it tells us that at that concentration, the cells are going to be dead. They're not going to undergo binary fission when you give them a nice nutrient-rich environment anymore. So those are all ways in which we can get important information about bacteria in a lab setting. But for the most part, those are just not practical in a clinical setting. When we have a patient who has a bacterial infection and we want to know the best type of antibiotic to use, if we don't know the bacteria, well, we can't make assumptions about the antibiotics. So honestly, we need to test a whole lot of different antibiotics. And we can do that using an automated test that is like the one that is set up down here. Okay? So this is all done on a very small scale. This is roughly like real life size. These results we can get within a few hours, and it's because we don't need our cells to grow up and divide enough for us to see them. This media has lots of different antibiotics that are being tested at the same time, and the media is formulated with a pH indicator, so it will change color when our cells are growing and producing acidic byproducts. So it's much, much faster. This is very similar to our tube serial dilutions, but we're just doing it on a much smaller scale using multiple antibiotics simultaneously. So we've got like ampicillin, tetracycline. I'm not interested in you knowing all of these different names that are up here. We have a couple of them that are repeats. But the idea here is that in the first well, we're going to have no antibiotic. So we use this essentially as a control to show, hey, yeah, we have bacteria here and they can in fact grow in this media. If we don't see growth here, that tells us that something has gone wrong and we need to dismiss our results. But we see here that we have growth in all of the antibiotic-free media. So ampicillin is going to be what is in all of these wells on the far left. Tetracycline will be the one that is in all of the next cells. So here we have our highest concentrations, and they decrease up until we get to the point where we have our lowest concentrations. Well, we see that ampicillin is not particularly effective against this particular bacteria. Tetracycline is at this particular concentration. And we can go on and we can look at a variety of different antibiotics all at the same time. So again, we're looking at all of this stuff out in a lab and performing these experiments to identify what our minimum inhibitory concentration is, which antibiotics that are going to be best selected for the pathogen that we're trying to kill. But you know what? At the end of the day, the pathogen and the antibiotic that you're going to use to inhibit its growth is ultimately all going to be within the human body. And so we have to consider human physiology when we are trying to dose antibiotics appropriately. So your kidneys and your liver, their job is to filter out substances that are within the body. So sometimes these substances are made by your own natural metabolic processes, but you know what? If you take any drug, your kidneys and your liver are going to indiscriminately filter it out as well. That's what they do. They're really good at their job. So when you treat a patient, you have to consider the physiology of their body. And we have to maintain a concentration that is sufficiently above that MIC in order for that drug to be effective in fighting off that patient's infection. So pharmacists can kind of go at this in a couple of different ways. They can have a single high dose of an antibiotic that is slow release. We may do multiple doses. If you've ever taken a round of antibiotics and you had to take one at breakfast, lunch, and then dinner, or maybe you had to take one in the morning and then when you went to bed. All of that dosing instruction comes down to this idea of ensuring a high enough concentration within the body to be effective. So when we have our diagram down here, we're looking at hours along the bottom, and then we're looking at concentration, blood concentration, so serum concentration of whatever drug it is that you are dealing with. We're going to see this same pattern regardless of drugs, but the microgram per milliliter may be different from one antibiotic to another, but the pattern is the same. We're going to see that when you take that drug and begin to absorb it, we see a nice high peak in that concentration, but then your kidneys and your liver go to work filtering out and we lower that concentration. Well, we want to make sure that we maintain our concentration above that minimum inhibitory concentration. So where we see that concentration dip close to it, this is where we would take our second dose. Maybe it's at a different meal or maybe it's the next morning. But we want to make sure that it's high enough above that minimum inhibitory concentration. But it's a dance. We don't just want to give these really high doses of antibiotics because sometimes these drugs can have harmful side effects. Sometimes the drug itself can have harmful side effects to our cells. We try our best to ensure that they are selectively toxic, but sometimes if we are limited on the drugs that we can choose, we'll opt for one that maybe has some more dangerous side effects because that's the best one to fight off the pathogen that we have. Additionally, we want to be aware of the kind of opening up the possibility for opportunistic infections. So always those side effects are something that we have to be aware of. We can't just give these really high doses of our drug. So when we start talking about dosing of drugs, there's some levels of doses that we need to be aware of. So when we're talking about the therapeutic dose, this is the minimum dose per kilogram of the patient's body weight that is effective at inhibiting the bacteria. So the therapeutic dose is the minimum dose that we need to actually do something, to be of use. So when we start talking about that minimum inhibitory concentration, we need to be above that. But we don't just want to give these high levels of antibiotic because we have to consider the toxic dose of a drug as well. So the toxic dose is the maximum dose per kilogram of body weight, again, that can be tolerated by the patient. This may be before we see the likelihood of kind of harmful side effects or increased likelihood of opportunistic infections. Ideally, we will have a big difference between the therapeutic dose and the toxic dose. So we want to make sure that we're not ever accidentally coming up close to that toxic dose. So we consider the therapeutic index, which is the difference essentially from that toxic dose to that therapeutic dose. We want it to be a really wide difference. A wide difference gives us a really nice buffer, a nice window, so that, if a patient takes one dose too early, they're not going to accidentally bump up against that toxic dose. By having a nice wide difference between our therapeutic dose and our toxic dose, we help to make sure that our patient is safe, that the drug is going to be effective but not harmful. All right, it is with this slide that we are going to stop lecture video A. We will continue by looking at specific antibiotic drugs in part B.